Well, thank you very much. It's a pleasure to be here. I get the privilege of speaking last in this day of our symposium. At least it's not yet 6 p.m. as indicated in Professor Melchior's lecture this morning of the class in, when was it? And the last class was 6 to 7 p.m., so we're not that late yet. It's a pleasure to be here as well because the first of these pioneer plant breeding symposium is how it started, was at the University of Minnesota back in 2009, I believe. And in fact, the idea to have, I started the ball rolling by emailing Jeff Graham at Pioneer saying, Jeff, would you give us funding to have a student-run symposium just for 3 years? We think it's good for our students. Would you give us money to run it for 3 years? If it works, after 3 years, we'll ask you for more money. If it doesn't work, then you didn't lose too much. But never in my wildest dreams did I think that it would develop to what it is today. So again, it's a pleasure to be here after more than a decade to be at one of these symposia, but now across the ocean. I'm giving my talk today without any slides. And so please bear with me. I'm getting lazy as I get older, so no slides for me today. So, my task today, I was told, is to give an overview of what plant breeding might be in the next 200 years. The nice thing about predicting 200 years in advance is that you or I will no longer be around in 200 years. So you cannot tell me, Rex, you told us something that was completely wrong. So I have that total freedom to say anything I want to say today without any ramifications whatsoever. But what I'd like to do is then structure my talk with regards to the definition of plant breeding and see how the different aspects of this definition of plant breeding have changed in the past years and how they might change in the years to come. So I define plant breeding as the genetic improvement of plants for human benefit. Plant breeding is the genetic improvement of plants for human benefit. So what I'd like to do is dissect this definition bit by bit and focus on the different aspects of the genetic improvement of plants for human benefit. Let's talk about humans. Back in 1800, the world population was 1 billion people. There were about 1 billion people in the year 1800 or 1818 when the University of Hohenheim was founded. Today, there are about 7 billion people right now. It's projected to increase to close to 10 billion people by the year 2050. And in 200 years, as shown earlier today, there are high projections, there are low projections. Most go with the middle projection. And the middle projection is after this steep increase in the world population, that the population will actually stabilize. The population will stabilize to about 11 billion people in the next 200 years or so. And so on the one hand, you may say, well, that's good to know. The world population is stable, but 11 billion people on this planet is still a lot of people. And obviously, with 11 billion people, these are 11 billion people who all have needs, and for whom plants would have to continue to provide benefits. And so, genetic improvement of plants for human benefit. What are these benefits that we are talking about that plant breeding has to address? Obviously, if you look back 200 years or so, the main benefits that would come to mind from those years and today would be food for humans and feed for the animals that we as humans eat. Food and feed. Those are the main two basic needs that were present for plants met in the past. Now you could add fiber, meaning fiber for clothing, food, feed, and fiber, basic needs. And we think of more recently, what are the more recent human benefits that breeders have sought to breed for? You could add fuel. Here in Germany, for example, in the recent past, biofuels have been a big thing. I think of in the University of Minnesota, back when fuel prices, not the university, but in the state of Minnesota, when fuel prices were high, we were talking about corn stoves. And I said, what's a corn stove? How can you make a stove out of corn? No, a corn stove is one that uses corn as fuel. You're burning corn just because corn was so cheap and fuel was so expensive. And you may say, well, I have a fundamental problem with just burning a feedstuff. But you could also argue, well, heat is a human need. In fact, you could survive far longer without food than without any heat at all. I'm glad that that situation has now changed, but still, the fact of the matter is, fuel will continue to be a pressing need in the future, and I would think that we could expect that breeding plants to meet fuel needs of humans will continue to be an important need in the future. Now, another aspect, food, feed, fiber, fuel, fun. In the past, perhaps there wasn't much breeding of plants for fun. But have you thought about the grass in the golf courses? Do you think that the grass in the golf courses just came from any grass? No. There was a lot of breeding effort that went into that grass so that the spin of the golf ball will be whatever it is, it'll be as uniform as possible. You could think of breeding for flowers. It's for fun, it gives us pleasure. It gives us aesthetic benefits and so forth. So you have that as a human need, and as we become richer and richer, as society becomes richer and richer, we could expect that to continue. But we could also think of what might be emerging human needs, human benefits that we would expect from plants as we look 200 years into the future. I'm speculating here, but let me mention a few. One is ecosystem services or ecological services. For example, in this day and age, we begin to hear about the carbon footprints that we have as humans. And so, for example, back in a decade ago or so, we had a project proposal. It came close to getting funded, but it did not. But for example, can we breed crops to mitigate for carbon sequestration? Can we breed crops to sequester carbon and keep it for a long time, sequestered? How might that happen? Well, one way is you could imagine, can we breed corn so that the roots are high in lignin, very high in lignin, and so that the carbon then is stored in that lignin under the soil for a long period of time. It doesn't decompose right away, and it keeps that as a carbon sink. So that is just one example. Ecological services. We think of ecological services at the University of Minnesota also in terms of cover cropping. Cover cropping to preserve the soil surface. So at the University of Minnesota, because it is extremely cold, what happens is that the soil is bare in the early spring. And so the idea then is to plant something. It's either an annual crop planted the season before, or a perennial crop that would provide cover for that bare soil, so that when we now plant our cash crops, corn and soybean, it's not in bare soil, and so therefore we have a cover crop that provides ecological services. Another human benefit that I think could be important in the future, it's actually important today, but we don't talk too much about it. It's called convenience. Have you ever heard of convenience as a breeding objective? Now, there are, for example, fruits that are known as convenience foods. For example, banana is an example of a convenience food because it is easy to peel and you eat it right away. Or an apple, you just bite into it. However, if it's a fruit that it's difficult to peel or takes a long time to process, well, that's not a convenience kind of fruit. And so, if we think of in the future, things like the internet and smartphones and watching TV and so on, we would be demanding more of this convenience type of aspects from the plants that we have. Just to give a few examples, two examples, and the convenience could be from the producer's perspective or from the consumer's perspective. In Japan, for example, they have developed not genetically, but by other means, watermelons that are shaped in cubes. Why would you like watermelon shaped in cubes? Well, you would store it more easily in your refrigerator. And when you cut it, instead of cutting something that's round, you just cut that cube and it's all squares and it's a lot more easier. And so, for example, you could speculate, could there be more things like this, where you develop something that's mainly for convenience, like breeding for shape, for example. From a producer's perspective, there could be things that would make production and harvest easier. The most difficult crop that I've ever pollinated is coconut. Anybody pollinated coconut here? Nobody. Pollinating coconut is a pain. I'll tell you why. First of all, you have to climb that thing. And in the Philippines, where I grew up, we did not have ladders. But what we had was we had that trunk, and you cut a wedge on the trunk. And you cut a wedge here and a wedge here, so you could climb that coconut like that, kind of like Spider-Man. And you have your gear with you when you sit on that coconut and you try to pollinate that, and then you do that for several days because they're not already on the same day. Harvest is also a pain because you need to climb that thing to actually get it and harvest it. And so one convenience item, if you're into coconut breeding and genetics, is just making it easier to harvest. Would it be possible to make the coconut so that it's senescence when it's a certain stage? Not yet full maturity, because you might not want full maturity, but at the right stage, the coconut just drops. And then you pick it up, and that's your crop. So that's an example then of a convenience type of breeding objective. Nutrition. Increased nutritional qualities could be it's an important breeding objective today. It could be even more important in the future. At Minnesota, for example, we have a new plant protein innovation center. The idea is to focus more on plants as sources of proteins rather than just trying to rely on animals as a source of protein. So, genetic improvement of plants for human benefit. Humans, we talk about benefits. How about the actual plants that provide these benefits? If you think of, again, I'm speaking from a US perspective, soybean is the second biggest crop in the United States, right after corn. Soybean was virtually inexistent in the US in 1818. It was present, but it was virtually unknown. And it is astounding to see, in actually less than a span of 150 years, to see soybean develop from basically nothing to the second major crop in the United States. Why? Because it was well adapted and it met needs for oil, it met needs for protein for animals. And so we can speculate, well, what are the new plants that might be present 200 years from now that we don't have today? And I have no answers for you, really. I don't know what it is. I'll just say, well, it's something like soybean, but it's not soybean, and we'll find it out. We'll find that out later on, right? But again, there could be different human benefits. You think of rubber, for example. Rubber, if you're a rubber breeder, you need to have a lot of patience. The generation interval for the rubber tree is about 40 years. So you need to wait 40 years for a breeding cycle. And so there have been interest in developing alternative crops to provide latex for rubber. Guayule, for example, is one example. And so, we need to think of what these human benefits are and what might be these new crops, either existing crops today or crops that are undomesticated but then can be domesticated to begin to provide these human benefits. Now, if we're talking about the genetic improvement of plants for human benefit, if we're saying genetic improvement that recognizes that there also is improvement that is not genetic. There is improvement that is not genetic, that's improvement that is primarily cultural in nature. And if we think of just developments in the last 200 years or so on, there have been changes in the way that crops are grown. For example, plant population densities today are certainly far greater than the plant population densities of yesterday. Rates of nitrogen fertilization are much greater today than they were of yesterday. And so we need to think of and we need to anticipate what these changes are in the environment and breed for them in advance. For example, if we say that there would be a higher CO2 concentration in the atmosphere in the future, is that something that we need to breed far in advance? Or are we saying, well, that change will be gradual anyway, and as the years go by, we'll be breeding for that? But it's interesting to look into the past because it can dictate what would happen in the future. For example, if you think of the two things that I mentioned, plant population density and nitrogen response, corn or maize is a prime example. At the University of Minnesota, we experimented with having what we call this dwarf corn as an alternative to the regular corn that we have. So dwarf corn grows about this tall. It is grown in spacings of about that wide instead of that wide. And so basically you're growing it like a small grain. And the idea being, well, if we radically change the plant population density, is that a new way of growing the corn crop in the future? The ecological benefit would be they're closely spaced, and therefore the roots provide more, they hold more of the soil. And we figured, well, if you're a seed company, you can sell more seeds with dwarf corn because they're at a higher plant population density. It turns out that that idea did not really work because the dwarf corn had a far, at least the germplasm we had, was of a far different maturity than the regular corn. It was so much earlier that it couldn't really compete yield-wise with the regular corn. But then it led us to asking questions, will it fit a certain crop rotation pattern? For example, if a farmer is harvesting the corn in June, can we plant this dwarf corn in mid-June or even July, and then harvest it before the snow comes by mid-October? So, again, the context there is we can change the environment in which the plants are grown, but as breeders, we are primarily interested in the genetic improvement, the genetic aspect. And for me, I've considered myself fortunate in having developed my career in the last 3 decades, because there have been a lot of changes in the tools that we use for plant breeding. My father was educated and trained at North Carolina State University and taught in cytogenetics. And when I chat with him, he fondly mentions the tools of his day, which were induced polyploidy and induced mutation, mainly with EMS or physical agents. And it was nice to see, to hear this morning of the contributions from induced mutation breeding. Now, when I started my career, quantitative genetics was my interest, and it was one of the main subjects that was being taught. But when I started breeding, it was simple. You crossed good by good and hoped for the best, and all selection was phenotypic. We evaluated plants in the field and chose the ones that were the best in the field. And I've told this story many times, but my life changed when one day a fellow graduate student from Spain walked into our office and told us about RFLPs, or restriction fragment length polymorphisms. And we said, what is this thing? And he was explaining to us what this can do. And my first real exposure to RFLPs was when we were asked to go to a cornfield to collect leaf samples for DNA testing. We were asked to wear gloves because our DNA, we were told, might contaminate the corn DNA. So we wore gloves. We were told to cut leaves that long for RFLP analysis. So we were chopping basically corn plants. Then we were told, okay, you fold those leaves, put them in a mesh bag and put them in a cooler, which we did. And then that cooler was driven off to the local airport, where there was a chartered airplane waiting to take those leaf samples to a lab in Iowa so that precious RFLP analysis could be done. And I go, my goodness, that is an expensive technology and something good better come out of this. But my advice to Tony Rex, think of when RFLPs are not as expensive as they are today, which was about $5 or so a data point, to when they cost the same as a paper clip that we use for putting the bag for pollinating. And I said, no, paper clip, how can that be? That's never going to happen. But that's where we are today. The cost of a data point now is about the cost of a paper clip. And so the question is, how do we use this tool in plant breeding? And throughout the years, we have seen how this has evolved, and we've heard presentations today on QTL mapping. QTL mapping, when to use it, when not to use it. And it was wonderful to see examples of QTL mapping for major QTL, because that's really where the procedure is most useful, is if you could map QTL, a major QTL with large effects, from a distinct source that confers either resistance to an abiotic or biotic stress. That has been found useful, no question about it. Another tool that has come that I was exposed to was association mapping, which is still useful today. My advice to you as breeders normally is that you shouldn't use association mapping. If that's your thesis, do that as well as you can, but for the most part, association mapping for a breeder is not very useful. The reason is that association mapping is designed to detect variation that is common. Breeders, on the other hand, are interested in variation that usually is rare. If it's common variation, yes, association mapping will detect it, but it's already there. What you're looking for the most part, is variation that is rare, and association mapping cannot detect rare variation. If I'm looking for rare, a disease resistance that is rare, and I have a mapping panel of 1,000 susceptible lines and 1 resistant line, I'm not going to find that association in a 1 to 999 ratio. But if I cross that resistant line with a susceptible line, now my allele frequencies are 50-50, that variation is not rare in that cross, and therefore I can do my QTL mapping approach. So I think that's a learning that we've had. Other tools that have come through my career, one is genomic prediction or genome-wide prediction, as I prefer to call it. And it's again the use of a large number of random molecular markers found throughout the genome to predict performance for different traits of interest. And in the last decade or so, we can look back and we can see instances, and we know instances in which it's useful and instances in which it is not useful. For example, it is most useful when phenotypic selection is ineffective. So if we can identify those stages in the breeding program when phenotypic selection is ineffective, that's when we would get the most value from these genome-wide predictions. We know that relatedness between the training population and the test population is key. We know that the higher heritability, the more accurate the predictions are, the more markers are, the better. And the larger the training population is, in general, the better the predictions are. It has worked well. It doesn't always work, but it works well on average, that it has now become, again, routine in most of the major species. My favorite example of genomic prediction actually is not in plants, but it's in animals, in dairy cattle breeding. They've used this procedure very aggressively, and a few years back, they predicted the best dairy bull, Holstein dairy bull in the entire United States, before this bull produced any progeny. The bull's name was Badger Fluff Fanny Freddy. Badger Fluff Fanny Freddy was predicted to be the best bull, and in fact, when the progeny of Badger Fluff Fanny Freddy came, those predictions were validated, because Badger Fluff Fanny Freddy's milk yield of the progeny was far higher than what has been seen in the past. Badger Fluff Fanny Freddy is no longer the top bull in the US. There's been a new top bull, but I don't think the name of this new top bull is as nice as Badger Fluff Fanny Freddy, which I really like. So again, we've moved genetic improvement of plants for human benefit, where we're moving through different tools that we encounter. And there are newer tools that have been mentioned in the symposium. Phenomics is one of them. The use of remote sensing, of robots, of data mining, data science to come up with phenotypic measurements. We've heard about genome editing today, which is a wonderful, wonderful tool that we have if we are allowed to use it. And so, but back to where we are. Okay, this is the now. Where does that leave us? What's the next 200 years going to look like in terms of genetic improvement of plants for human benefit? And I've thought about this for quite a while, and how do I summarize what I think breeding in the future might look like? And my summary is simply this. I think breeding in the next 200 years will be more precise. Breeding in the next 200 years will be more precise than it has ever been. And this level of precision comes from many sources, and it comes in different forms. I mentioned to you that when I started my career, breeding was simple. You evaluated plants in the field, chose good by good, crossed the two, and just relied on random progeny, and you selected the best, and then you repeated that process again. And now we have a lot more tools at our disposal, and we will have a lot more tools at our disposal to potentially make it more precise, but we need to know how to use these tools to make the breeding process more precise. I like this, to illustrate this point further, I like this analogy of how plant breeding is different from car design. So so the way cars are designed, is you take your knowledge of how cars work, and you synthesize a prototype, and you build that, and you test it, and you keep refining that process. And I would say that if a plant breeder were to design cars in the way that the plant breeder breeds plants, here's how it would be. A breeder would take two high-performance cars, like a Porsche or a Ferrari and a Lamborghini. You would exchange the different parts of these cars into a mosaic. Could be the engine from the Ferrari, one wheel from the Lamborghini, muffler from the Ferrari, and so forth. You find the best mosaic and you repeat that process. Why? Because it's a biological organism for which we do not have much control right now. I mean, we can control which cars we cross, we can control that quality control process if you would to pick the best ones, but so far we have not had that precision yet in the entirety of the breeding process. And we heard ideas today of how that might change, and I'm actually investigating that at the University of Minnesota right now, and it's called targeted recombination. Targeted recombination meaning the ability to either induce or select for recombination where the breeder wants that to be. And so how do you do that? Well, first of all, you need to know where recombination should be. And we have ways to estimate that, because now we can estimate the effects of markers along the entire genome. So we can take chromosome 1, for example, and let's say chromosome 1 is 100 markers, okay? And from marker 1 to 25, the good allele is mostly in the first parent. And from marker 26 to 100, the good allele is mainly in the second parent. So we say, well, recombination should be between marker 25 and 26, because that would maximize performance, and we pick the good homologue. And we do that for each chromosome. And then through a process like marker-assisted backcrossing, we pyramid as many of those recombinations as possible into one line. And again, it's something that I'm investigating right now. But the whole idea, regardless of whether that works or does not work, is the idea of having the tools to make breeding more precise. It's either at the genetic level, saying where targeted recombination should be. It could be an interplay with the environmental level, a better characterization of the environments that we have, so that we can have better placement of the products we have to specific environments. And so, again, as we look back at the last 200 years and focus, try to speculate in the next 200 years, I'd say that we can make educated speculations, but a lot of things are unknown. A lot of things are unknown, and part of the joy is being in that journey of discovering these new things in our field. And these new things lead to new things, and lead to new things, and lead to new things. And so as PhD students, you're in this wonderful journey of this career in the genetic improvement of plants for human benefit. You're developing stuff that has not existed before. Isn't that exciting? You have a new plant in the field, and you can say to yourself, this plant that I developed, mainly, well, you didn't really do too much. You just crossed 2 plants and harvested the seeds, right? But still, nevertheless, you had a hand in the creation of this unique plant individual. And you could begin to determine, well, what are the human benefits that this plant could meet? And so, to close, I'd like to close with a quote from a former defense secretary in the United States, Donald Rumsfeld. He said this during a press conference. And this is a bit of a paraphrase, but basically he said, there are known knowns. There are things we know we know. There are known unknowns. There are things we know that we do not know. And there are unknown unknowns. That is to say, there are things we do not know, we do not know. Let me expand on that. Take genes, the most basic factor that we work on, genes. If we were in Hohenheim in 1818, genes were an unknown unknown. We did not know that there were such things as genes. That was just, we don't know. It hasn't even entered our brains that we did not know that we did not know. Okay? Now, after the discovery of Mendel's laws, if we were living in, say now, in not 1818, but 1918, genes were unknown unknown. We knew there were things such as genes. We did not know what genes were made of. In fact, there was investigations, was it a nucleic acid or was it a protein? So that was unknown, unknown. And now we live in an age where genes are unknown, known. We know that genes are made out of DNA. And in fact, we know far beyond that. We know how to manipulate this DNA to come up with plants that meet human benefits. And so again, thanks for the students for organizing this, and I encourage you as we continue, as you embark and continue your careers, to continue to be learners. You are learners right now, and don't let your learning stop, but keep on learning. I ended the quote from my own advisor, and well, this is not from him, but he had this posted in his office. In times of change, learners inherit the earth, but the learned find themselves beautifully equipped to deal with a world that no longer exists. So if we stay at what we currently know, things around us will change, and we won't be able to function in that world that has now changed. So keep learning. Thanks.
Yes, thank you, Professor Bernardo, for this very inspiring talk. And we have time for a few questions. Maybe continue the speculation.
Alex, thank you very much for very motivated talk. Take it in account, and thank you very much specifically for vision 200 years ahead. Take it in account that 10 billion people will be, for example, and majority of them probably will not live in village, probably live in big capital city. What do you think about urban agriculture to produce? Probably we need already to start to think more seriously to produce a plant which is grown balcony to produce more, and to continue with convenience of the food. For me personally, the tomato I get picked from my garden tastes as much better as to get from the market. Probably the other argument we need to think on the future.
That's a great comment. And so certainly the university where I work, it's an interesting situation because it's a major university, but it is in a university of nearly 3 million people. So we study agriculture in an urban environment. And when I compare that to previous universities I've been with, it certainly is a different dynamic that we sense within the university. And so there are issues that we deal with in an urban setting that we never had to deal with in a rural setting. Obviously, urban agriculture, local foods is one of them. But this whole question of local foods, well, first of all, I don't like local foods in the sense that I like my wine that came from France, or I like my, if I were to survive on only local food, I won't be very happy. So, but could that be a breeding objective? Because post-harvest quality, could that be a breeding objective? So you're saying that in the grocery store, you could taste a difference between an item you pick from the grocery store and an item you pick fresh from your garden. Would that be a meaningful breeding objective? Post-harvest taste and quality, so that that quality stays and it tastes as good. But certainly with the urbanization and the, when you're poor, you just eat something. And I grew up in the Philippines where the food budget was about 60% of what you made. Food was 60%. I moved to the US. I go, food here is too cheap. Amazing. So cheap. And so now when you have choice, when you have that ability to have choice, then you want to choose, you become more specific. You become more picky, if you would. And so I think we could expect more picky consumers in the future. And so that could be a need that would need to breed for. Any further questions?
Yes, thank you very much for the talk. My question is about the plant genetic resources. Looking back the last 200 years, do we still have the same foundation? Like we improved our tools, but do we still have the same foundation in terms of genetics, like amount of plant genetic resources?
Thank you. I'm very glad you asked that question. So just a little secret, the nice thing about not having slides is if you forget something, nobody knows. But I confess I meant to mention that, but I just forgot. But plant genetic resources, if you think, I think of breeding as like a three-legged stool, where the seat is successful or useful cultivars, and you need three legs. You need breeders, you need tools and methods and resources is the second leg, and the third leg is your germplasm. Without any of those, you won't be successful. And so I think we are in the time and age in which most of the breeding organizations they have well-educated, well-trained people. They're using the latest and greatest tools and methods. What would differentiate them is the germplasm that they have access to. And so I think there certainly has to be more attention paid to how do we better utilize the genetic resources that we have. Let me give you an example that we're working on from our work on targeted recombination. A focus on utilizing exotic germplasm in the past has been on finding major QTL or QTL found in exotic germplasm and introgressing one or a few QTL at a time from exotic germplasm into elite germplasm. With genome-wide marker effects, what the question we're looking into is not introgressing one QTL at a time, but introgressing segments of the genome at a time from the exotic source to an elite source, knowing that just one of these markers might not have a large effect, but if we perhaps introgress this segment of the chromosome, would that have enough of an effect to make a difference? So again, I think we need to explore as breeders these types of approaches of using the modern tools that we have to better capitalize or exploit the diverse wonderful germplasm resources that we have.